Morning. I would like to thank the International Electronic Conference on Nutrients chairs for the invite to present our work in the conference. The topic of my presentation is the interactions between the mechanistic target of rapamycin and the microbiome. So what I would like to do is to go over background of what's Mtor, how it relates to the metabolism and the microbiome. And then I'll present our work in vivo data on Mtor in orthotopic mouse model, where I will look at the cell signaling, tumor growth, and metabolomics. Then I will present the data in vitro in pancreatic cell culture work. And there I will look at the cell cycle, metabolomics, and mitochondrial function. And then I'll end up by conclusions. So what's Mtor? Mtor stands for the mammalian target of rapamycin. It is a PI3 kinase-related protein, but it's actually a serine threonine kinase. It's a large protein with a size of 289 kilodalton, and it's a large protein which have different binding domains. From the N-terminus, there are several heat repeats, and then there is the fat domain, and then the FRB binding domain, which binds to the drug rapamycin. There is adjacent to it is another kinase domain, which is blocked by ATP competitive inhibitors. And it's a kinase, so it phosphorylates itself by autophosphorylation at the site serine 2481, close to the C-terminus, and then it phosphorylates downstream targets. This Mtor is part of several signaling pathways, and it is dysregulated in chronic diseases, including insulin resistance, cardiometabolic disease, diabetes, and cancer. It integrates signals from nutrients, from energy and environmental cues, and then transmits the signals to downstream targets. It functions by having two distinct complexes named mTORC1 and mTORC2. These two complexes are located in every cell, and the mTORC1 complex is nucleated by mTOR protein and other proteins, including specifically Raptor, which is exclusive to mTORC1, and then additional protein partners, such as the MLS8 and Pres40 and Deptor. The other complex, which is mTORC2, it binds exclusively to the protein Rector, in addition to some shared proteins with mTORC1. While a lot of the upstream signaling of mTORC1 is recognized, so it's an energy sensor, it senses low energy, and is also activated by several nutrients and amino acids, and it's downstream of the insulin signaling pathway. So several of the upstream regulators of mTORC1 has been elucidated, while mTORC2 is only regulated by growth factors, and not much is known about the upstream signaling of mTORC2. So as I mentioned, mTORC1 is a nutrient sensing pathway. It receives signals from all and inputs from all the macronutrients. So when carbohydrate in product is glucose, and proteins are broken down to amino acids, and lipids generate energy and ATP and AMP, they all are integrated by the mTORC1 complex. And mTOR in turn regulates synthesis and growth. Also, it regulates the metabolism. It's an anabolic complex, and also the gut microbiome. So if there is dysregulation of mTORC1, If there is problems with the synthesis with S6K and for EBP translational proteins, this will lead to problems with cancer. And also, if dysregulated, there is a metabolism leading to insulin resistance, diabetes, and obesity. So the rationale of this work is that given the pleiotropic effects of Mtor and mTORC1 and mTORC2, and mTORC1 effects on nutrient sensing and metabolism, as well as immune response. As I mentioned, there's proposed interactions between Mtor and the gut microbiome. So we hypothesized that Mtor has crosstalk with the gut microbiome to modulate nutrient metabolism. So in the following slides, I will summarize the recent advances in the microbiome research. Then I will present our work on mTORC1 and mTORC2 metabolism in the pancreas. The NIH Human Microbiome Project has been carried out over 10 years, and there are two phases of the integrative human microbiome. The first phase that looked at healthy cohort studies and isolated the microbiomes from different conditions, and led to a lot of resources that's available and data available and tools and protocols to study the human microbiome. It was cross-sectional, so they looked at the community composition of the microbiome, the microbial pathways, the host genome sequence, and the microbial isolates. And then the phase 2 of the Human Microbiome Project looked at the disease states, such as the inflammatory bowel disease and pre-diabetes and diabetes, as well as preterm birth. It took a longitudinal sampling and characterized the host and the microbiome, again, the community composition, microbial pathways and virome, as well as looked at the big data in metatranscriptomics, metaproteomics, and metabolomics. And recently, Mtor has been shown to interact with the microbiome in a paper published by Yang et al. In Natural Communications. They have shown in the CD3T cells, CG4T cells, that the phospho-Mtor regulates the microbiome and short-chain fatty acids, as well as the butyrate. So these interactions with the microbiome inspired us. So what I'm going to present is overview of Mtor. As I mentioned, it interacts with several pathways, and it has already crosstalk with several pathways. So it's located downstream of the insulin receptor pathway, and therefore mTORC1 is regulated under insulin receptor, PI3 kinase AKT, which inhibits the TSC1 and 2. That's inhibitory to the Reb-GTP, which activates mTORC1. In addition, mTORC2 complex phosphorylates AKT at serine 473, and therefore it activates it, and if it's dysregulated, it can lead to cancer as well. Mtor also has a crosstalk with the RAS-MEK pathway, and it also receives signals directly from amino acid receptors and transporters through the RAG complexes, and also has immune microenvironment, interacts with the cytokines and CD4. So given that it's a pathway that's involved in nutrient signaling, and there is crosstalk between mTORC1 and mTORC2, as well as other crosstalk with other pathways that are involved in metabolism, and it's also important in the immune response. We set about to investigate the mTOR in metabolism and the microbiome. So in the next few slides, I will summarize our data, in vivo data, where we looked at an orthotopic mouse model of pancreatic ductal adenocarcinoma and looked at the cell signaling, the tumor growth, and metabolomics. So the rationale for the study was that mTORC1 mediated amino acid sensing and is utilized by cancer cells to drive the metabolic reprogramming. Further, cancer cells repurposes the nutrient signaling capacity of mTOR for metabolic reprogramming to drive cancer progression. And recently, a new study has suggested that mTORC2 may be a glucose sensor in cancer metabolism. So given that, in our study, we investigated the impact of inhibition of both complexes mTORC1 and mTORC2 by drugs and the synergism with the anti-diabetic drug metformin, and looked at the pancreatic tumor growth and metabolomics. So the study design was that we took advantage of the availability of triple transgenic mice. These are having triple mutations of the KRAS, P53, and PDX1 pancreatic progenitors, and these known as the KPC mice, they spontaneously develop pancreatic tumors due to the triple mutations that drive the metabolic reprogramming of cancer cells, and they develop spontaneous pancreatic tumors. So we took cell lines from the tumors from these KPC mice, and we implanted them intrapancreatically in albino mice. We followed them by checking the tumor after the injection. We checked the tumor in several days until the tumor within 2 weeks or 14 days, the development of the tumors in the pancreas. Then we injected the albino mice daily intraperitoneally with different combinations of drugs. First, the vehicle, which was the DMSO control, and then a group of taurin-2, which blocks mTORC1 and mTORC2. We used a low dose of taurin-2 and a high dose of taurin-2, as well as the metformin, and then a combination between taurin-2 and metformin. We injected them intraperitoneally for 12 days, and then we sacrificed the animals. So we looked at the cell signaling, and as expected, when we had taurin-2, which blocks the kinase domain of mTOR, it's an ATP competitive inhibitor, and therefore we looked at the phosphorylation of AKT at serine 473, and it was decreased by both the concentrations of taurin high and taurin low compared to the total AKT in the lower lane. We also looked at the phosphorylation of S6, which declares that there's an mTORC1, and again with taurin-2, both high dose and low dose, it inhibited the phosphorylation of S6. So this clarified that our injections led to the inhibition of both complexes mTORC1 and mTORC2, and it also inhibited the autophosphorylation or reduced the autophosphorylation of mTOR at serine 2481. Then after we sacrificed the animals, we looked at the tumors, and the tumor size was decreased significantly with the combination of taurin-2 and metformin, both concentrations. And this was significant for the tumor volume that has significantly decreased with the combined combination of taurin and metformin. as well as the tumor weight was also significantly decreased. And the maximum effect was the synergism between Taurine-2 and metformin, and to a lesser extent, metformin alone, also lesser extent, the Taurine-2 low concentration and high concentration. Then we looked at the metabolomics, and using the partial least square discriminant analysis, and we were able to find that the control, the control is significantly different than the combination of taurine and metformin. And this affected all the metabolism of both the glycolysis and TCA cycle, as well as several pathways. So we deconvoluted the pathways using the MetaboAnalyst programming, and we found that the combination of taurine and metformin significantly affected amino acid metabolism. Particularly, there was significant elevation of the phenylalanine concentration, almost fourfold increase in phenylalanine concentration. And there is also increase in the nucleotide metabolism in pyrimidine, and uracil was significantly increased with the combination of treatment of taurine-2 and metformin. On the other hand, we found that for glucose and hexoses, both glucose and galactose were significantly decreased with taurine and metformin. So as we looked at the functional enrichment data, we found that phenylalanine metabolism was significantly increased with the combination of metformin and taurine. Also, hexose metabolism has been significantly different, as well as the pyrimidine metabolism. Then we also looked at the short chain fatty acids, and we found that for the phenylpropiolic acid, there is significant decrease of the concentration with all treatment with taurine and metformin combination, with metformin and with taurine high and low concentrations compared to the control, suggesting that all treatment altered the short chain fatty acids which interact with the microbiome. And there is another short chain fatty acid, the isobutyl glycerol or glycerin, and it has significantly increased with all the concentration. So that suggests that the short chain fatty acid has been altered in both directions. Some of the short chain fatty acid has increased and some are decreased. So in conclusion, the combination of Mtor dual inhibition by taurine-2 and metformin is associated with an altered metabolomic profile and short chain fatty acid profile, which may alter the microbiome. And a significant reduction of the pancreatic tumor burden has been shown with inhibition of mTORC1 and mTORC2 compared with a single agent therapy, and it is better tolerated when mTORC1 and mTORC2 inhibition together with the metformin administration. Both of them together were better than the single agent therapy. Next, we looked at the in vitro data in cell culture in pancreatic cell lines, and we investigated the cell cycle, metabolomics, as well as the mitochondrial functions. So we were intrigued by the data we obtained from the mice model, and we know that the gut microbiome is altered in type 2 diabetes and in pancreatic cancer. We also know that PDAC, or pancreatic ductal adenocarcinoma, and the diabetes have bidirectional relationship. So type 2 diabetes and new onset diabetes have been shown 1 to 3 years before the diagnosis of pancreatic ductal adenocarcinoma. And the majority of pancreatic ductal adenocarcinoma patients are hyperglycemic. About 50% have concurrent diabetes, and the hyperglycemic diabetic state in the patients with pancreatic ductal adenocarcinoma enhances the oncogenic growth and tumor dissemination. So we set out to investigate the mechanistic pathways in cell culture. The recent reports that indicated that 50% of the pancreatic ductal adenocarcinoma patients are diabetic at the time of diagnosis, and that the anti-diabetic drug metformin, which indirectly inhibits Mtor, has emerged as a potential therapeutic target for PDAC. Therefore, the objective of our study was to determine the metabolomics changes, mitochondrial function, short chain fatty acid profile in pancreatic cell lines by inhibiting Mtor and with synergism with metformin. So we looked first at the signaling, cell signaling, to verify that we inhibited mTORC1 and mTORC2. So once again, we looked at the phosphorylation of AKT, and it's inhibited by taurine, but not by metformin. And then the combination of taurine and metformin blocked the phosphorylation and activation of AKT at serine 473. Similarly, for mTORC1, phospho S6 was inhibited with taurine, and that compared to the total S6. We looked at the cell cycle, and as mTOR has an effect in cell growth and proliferation in cell cycle, so taurin-2, which inhibits both mTORC1 and mTORC2, led to increase the apoptosis, and also led to inhibition or decrease in the G1 and G2 phase. When we looked at the metabolomics, we found that in HbF2 cells, the glycolysis intermediates, such as the fructose 1,6 bisphosphate, was decreased with treatments with taurin-2 and metformin and the combination of taurine and metformin. Also, the NAD concentration, which is the energy equivalent, was also decreased by taurin-2. And the 3PG was decreased, suggesting that several intermediates of glycolysis has been inhibited by mTOR1 and 2 inhibitors in HbF2 pancreatic ductal adenocarcinoma cell lines. Similarly, we found similar effects that the inhibition of by taurin-2 and metformin decreased the TCA cycle intermediates, such as the acetyl-CoA concentration, citrate and isocitrate concentration, as well as malate, and the FAD, which is the energy equivalent, was decreased by the treatment, and there is decrease in ATP concentration. So we wanted to look at the mitochondrial functions using the live cell bioenergetics, using the Seahorse XF analyzer. And basically, it allows the studying both complexes, which processes for energy, that is the oxygen consumption, which determines the mitochondrial concentration. So the oxygen consumption rate, or the OCTR, as well as the protons that are released, so to determine the glycolysis when independent of oxygen. And this is the glycolysis conversion of glucose to pyruvate and then lactate. And the release of lactate leads to acidification of the media surrounding the cell. So we measure the extracellular esterification rate, or ECAR, and that's both ECAR and OCR are measured simultaneously. So it will allow us to look at the mitochondrial respiration as well as the glycolysis and formation of lactate. So looking at the beta cell, we found that with this looking at the mitochondrial functions, that there is decrease in the OCR, the mitochondrial respiration, with all the Mtor inhibitors, taurin-2 and with metformin, and the combination of taurin-2 and metformin, as well as rapalink, which inhibits rapamycin binding site, as well as ATP inhibitor. And again, also, the treatments also led to the decrease of the esterification, which is the indication of the rapid glycolysis and lactate production. So as a reference of the mitochondrial respiration, so treatment with oligomycin, which inhibits complex 5, again, is associated with a decrease in the mitochondrial respiration in taurin-2 and metformin. And then the combination of treatment also had a higher impact compared to the control. And then using the uncoupler FCCP, which uncouples oxidative phosphorylation, so we were able to see the differences again between the control and the different treatment, and there was significant difference. There's significant reduction in the mitochondrial respiration. And there is also, after addition of the rotenone antimycin, which inhibits both complex 1 and complex 3, the difference was maintained, significant difference between the control and the treatments. And that's also was shown by inhibition or decrease in the ATP production with taurin-2, with metformin, and taurin-2 and metformin, rapamycin to a lesser extent. And there is also decrease in the proton leak. Similarly, maximal respiration and the spare respiratory capacity were also decreased compared to the control. So in conclusion, our in vitro data in cell culture suggests that mTOR inhibition by taurin-2 that inhibits complex 1 and complex 2 altered the cell cycle, and it altered glycolysis and TCA cycle, as well as mitochondrial functions, both the mitochondrial respiration and the rapid glycolysis and lactate production from esterification. Our in vivo data in mice showed that the mTOR inhibition that inhibited both mTOR1 and mTOR2 reduced tumor growth, and it altered the metabolomic profile, as well as the short-chain fatty acid in the orthotopic mouse model. These changes in the production in the short-chain fatty acid may alter the gut microbiome. And now the recent advances in the genomics and metabolomics has enabled identification of several microbiota species. And therefore, these links and these advances will help understanding the interactions between diet and nutrient composition and the gut microbiome interactions. Finally, the altered microbiome host interactions may play a significant role in the inflammation and CG4 and T subsets, as well as in chronic diseases and insulin resistance, cardiometabolic risk, diabetes, and cancer. Finally, I would like to acknowledge my collaborators at the University of Nebraska and at the City University of New York and the Advanced Science Research Center. And I thank you very much for listening. If you have any questions, my email is listed, and I look forward to hearing from you. Thank you.